The nature and amount of antigenic peptide can also affect the differentiation of CD4 T cells. Another factor that influences the differentiation of CD4 T cells into distinct effector subsets is the amount and exact sequence of the antigenic peptide that initiates the response. Large amounts of peptide that achieve a high density on the surface of antigen-presenting cells tend to stimulate Th1 cell responses, whereas low-density presentation tends to elicit Th2 cell responses. Moreover, peptides that interact strongly with T cell receptor tend to stimulate Th1-like responses, whereas peptides that bind weakly tend to stimulate Th2-like responses. This difference could be very important in several circumstances. For instance, allergy is caused by the production of immunoglobulin E antibody, which requires high levels of interleukin-4, but does not occur in the presence of interferon gamma, a potent inhibitor of interleukin-4-driven class switching to immunoglobulin E. Antigens that elicit immunoglobulin E-mediated allergy are generally delivered in minute doses, and they elicit Th2 cells that make interleukin-4 and no interferon gamma. It is also relevant that allergens do not elicit any of the known innate immune responses, which produce cytokines that tend to bias CD4 T cell differentiation toward Th1 cells. Finally, allergens are delivered to humans in minute doses across a thin mucosa, such as that of the lung. Something about this route of sensitization allows the host generators of Th1 responses, like Leishmania major, to induce Th2 responses. And that's the nature and amount of antigenic peptide can also affect the differentiation of CD4 T cells. Have a great day.